All right, so this is the third and final part of this week's lectures. In the previous portions of this week, we've talked quite a bit about the cells involved in the immune system. In this section, we're shifting our focus away from the cells themselves and looking at the substances that help those cells communicate and then carry out the immune response. And these are our soluble mediators. We're going to spend most of our time on two major groups: the complement proteins and cytokines. Then we'll connect those concepts back to the clinical laboratory by looking at complement testing in acute phase proteins, or you might also see it called acute phase reactants. Again, you do not need to memorize every protein involved in every single pathway. What I really want you to understand is how these systems work and what they accomplish, and what their laboratory results can tell us about a patient. So these three questions are essentially the roadmap for this entire lesson. So first, how does complement amplify immune defense? Second, how do cytokines allow immune cells to communicate with one another? And finally, because we're laboratorians, how can we actually see the evidence of these processes in laboratory results? So as we move through the chapter, keep coming back to these three questions. Again, the big idea is that soluble mediators aren't immune cells themselves. They're signals and proteins that allow the immune system response to become coordinated and amplified. So there are several objectives here, but notice how most of them ask you to compare, explain, describe, or interpret. That's intentional. I care a lot more about whether you can look at a complement pathway and explain what's happening than whether you can memorize a long list of complement proteins. By the end, you should also be looking, or you should also be able to take something like a decreased C3 or C4 result and start thinking about why that result might be decreased and what additional testing could help us. So that's the level of understanding that we're working toward ultimately. So again, don't try to memorize this entire list right now. Think of this slide as a vocabulary map for the lesson. On the complement side, some of the terms that we're going to keep coming back to are C3, C3A, C3b, opsonization, and the membrane attack complex. On the cytokine side, we'll distinguish some of the major families, including interleukins, interferons, TNF, colony stimulating factors, and chemokines. Then we'll finish with laboratory markers such as C-reactive protein or CRP and other acute phase reactants or acute phase proteins. By the end of this lesson, these terms should have a context rather than just being definitions. So this is probably the simplest way to think of soluble mediators. They're not just immune cells. They aren't immune cells. They help immune cells do their job. They can amplify an immune response. They can recruit cells to an area of infection or injury. They can promote inflammation, help remove immune complexes and give us measurable evidence that an inflammatory process is occurring. So, if our neutrophils, macrophages, lymphocytes, and other leukocytes are our workers, soluble mediators are a part of that communication and coordination system, telling those workers what needs to happen. So this slide really summarizes those functions as amplification, recruitment, inflammation, immune complex clearance, and laboratory evidence of disease activity. So first, we're going to start with complement. This section can look really overwhelming because there are a lot of proteins with numbers and letters attached to them. But please don't panic over the alphabet soup. We're going to build from the big picture first and then work our way into the individual pathways from there. So, complement is a group of proteins that circulate in the blood, primarily found in an inactive form. So, I like to explain complement as it's always in our bloodstream, right? It's always just kind of floating around, waiting to get that call to activation. So, the inactive state is what's important. We don't want complement constantly attacking cells or generating inflammation. That would be bad, right? When complement encounters the appropriate trigger, proteins become activated sequentially. That's why we call it a cascade. One protein activates another, which activates another, and then the response then becomes amplified. So remember this phrase: different triggers, shared cascade, immune effect. That idea will make the three pathways much easier to understand. So again, different triggers, shared cascade, immune effect. So there are three major ways that complement can get started. The classical pathway is associated primarily with antigen antibody complexes. The alternative pathway can be activated directly by microbial or foreign surfaces. And the mannose binding lectin pathway recognizes carbohydrate patterns, particularly mannose containing structures, on microorganisms. So the starting points are different, but here's the important part of it: they don't stay separate forever. All three pathways eventually converge at the activation of C3. So I want you to keep that in the back of your mind, right? So they start in different ways, but they all meet at that point of C3. So this diagram may look intimidating at first. So I don't want you to try to memorize every single thing. Instead, find C3. Find C3 in each pathway. Look at the alternative pathway. Now look at the classical. And then now look at the lectin. What happens at all three? Eventually we form C3. We form a C3 convertase, and C3 gets cleaved. And then that little C3A then goes on to the formation, and so on. So that's the organizational concept I really want you to remember. So the beginning tells us what activated complement. The middle converges around C3, and then the latter portion produces the specific effector functions of complement. So again, the diagram on this slide is showing that convergence around that C3. So why do we care about C3? So this slide is what I would say is one of your high yield slides. So C3 is the central turning point of complement. When C3 is cleaved, we get two things, right? We get C3A and C3B. And a simple way to remember their general roles is C3A promotes inflammation, and then C3B coats the target. So C3B acts as an opsonin, which makes the target easier for phagocytic cells to recognize and ingest. So when you see C3B, I want you to think opsonizations and phagocytosis. When you see C3A, think inflammatory signaling and recruitment. The easiest way to remember classical complement activation is antibody gets complement involved. Once antibody has bound to antigen, the C1 complex can recognize the antibody associated complex and then initiate that cascade. From there, we can eventually activate what? C3, and we can proceed towards the terminal complement pathway. But again, don't worry about memorizing every intermediate component right now. For at this moment, we'll start with classical is antigen antibody associated activation. Now, the alternative pathway is different because we do not need a specific antibody response first. Complement can interact directly with an appropriate either microbial or foreign surface. And that makes this pathway particularly important in the innate immunity. So remember, the adaptive immunity takes time to develop. The alternative pathway gives the body another mechanism for responding before highly specific antibody response is then established, right? So classical, antibody associated. Alternative, foreign surface, no specific antibody required. So the mannose binding lectin pathway, also known as the lectin pathway, or you might also see it referred to as the MBL pathway, either way. It's another innate mechanism. So instead of recognizing antibody, it recognizes patterns on microorganisms. Mannose binding lectin recognizes carbohydrate patterns that contain mannose and associated MASP enzymes initiate that complement cascade. Again, it is not antibody required. So the three starting points are, again, antibody, classical, foreign microbial surface, alternative, microbial carbohydrate pattern, lectin. Different trigger, same overall goal. So eventually the complement activation reaches that terminal pathway that forms the membrane attack complex, or the MAC. The MAC consists of our C5b through C9. These components then assemble in the target membrane and create a pore. So once its membrane integrity is disrupted, the cell can lyse. This is the most traumatic effect of complement. So it's often the one that students remember, but complement does so much more than kill cells, and that's what we'll look at next. So this is another this is another slide I would recommend to memorize or to get to know well. Rather than memorizing a list, remember 4 phrases: code it, call, clean it, kill it. Code it, call it, clean it, kill it. So code it. You could see how the opsonization, remember that C3b is what's going to code, right? Call for help, chemotaxis, inflammation. Then we're going to clean it up, remove the complex, and then kill it, cell lysis through that MAC. So again, code it means opsonization. Call it or call for help means inflammatory signaling and recruitment of immune cells. Clean it up refers to helping remove immune complexes. And then kill it refers to membrane damage and lysis through MAC. So if you understand those 4 functions, you understand why complement is clinically significant or clinically important. So now we're going to shift from immunology into the clinical laboratory. So why do we care? Why would we measure complement? So sometimes we're investigating whether the patient has a deficiency. Sometimes we're looking for evidence that complement is being consumed because the system is actively being used. And sometimes we're monitoring inflammatory or immune disease activity. So this is where interpretation really gets interesting. And honestly, this is a huge interest and thing that intrigues me personally. So decreased complement level does not automatically mean the patient can't produce complement. It could mean they're using it. So always ask, is this low because of deficiency or is it low because of consumption? Conversely, the complement proteins also behave as part of an inflammatory response. So increases occur in inflammatory states. So complement deficiency becomes particularly important when we see recurrent infections. So one association I want you to remember is recurrent Neisseria infections. If a patient repeatedly develops unusual or severe Neisseria infections, complement deficiency should at least enter the differential. This is where the patient history also becomes important. And a single laboratory value really isn't enough. We need to ask, what is the clinical pattern? What is the overall picture brought by the patient to testing in the first place? So rather than immediately measuring every single individual complement protein, we can start with functional pathway testing. CH50 evaluates that classical pathway, and then AH50 evaluates the alternative pathway. So, depending on the clinical question, we may also measure C3, C4, mannose binding lectin, or individual components. So, nice clinical example here is hereditary angioedema, where C1 inhibitor may be decreased or even dysfunctional. C4 is also commonly a part of the initial laboratory evaluation as well. And then one clinical clue is angioedema without urticaria. So this is a good example of combining the patient's presentation with targeted complement rather than ordering everything, throwing everything at the wall and seeing what sticks. That's why our job as laboratorians is so important. So we're finished with our major complement concepts, and now we're going to move into our second major group of soluble mediators, which are our cytokines. With complement, think cascade and amplification. With cytokines, start thinking communication, coordination. So complement amplifies the response and cytokines coordinate it. Cytokines allow cells to communicate. They can tell another cell to activate, move toward an area, proliferate, differentiate, or even change its behavior. So we're moving from a protein cascade to something that functions more like an immune signaling network as a whole. So cytokines are generally produced in response to cell activation, and they bind to specific receptors on target cells. So, an important concept is that they don't all have to travel throughout the entire body. Some act on the same cell that secrete them, while others act on nearby cells. And cytokines do opposite things depending on the situation. Some promote inflammation, some suppress it. Some stimulate cell growth, some help regulate or even shut down a response. So don't think of cytokines as inflammatory chemicals. They're more regulators of the immune system. There are three concepts here that are worth understanding. Okay. The first is redundancy. Multiple cytokines may produce similar effects. The second is amplification. One cytokine can stimulate production or activity of additional cytokines. And the third is feedback. So, cytokine signaling can increase response, but it can also suppress or regulate it as well. So, that's part of why the immune responses are so complex. It isn't one molecule turning one switch on or one switch off. Again, it's that network of interacting signals. So that brings us to our interleukins. And interleukins are a large group of cytokines that are involved in communication among leukocytes and other cells. Different interleukins have different functions. So, again, I absolutely do not expect you to memorize every interleukin number in this section. Instead, it's really important to understand their general role, and this should also help you in hematology as well. So, interleukins help regulate things such as cell growth, differentiation, movement, activation, and inflammation. You'll see in hematology that you'll start with a pluripotent stem cell, and depending on the specific interleukins, that helps it differentiate. So later, when you encounter individual interleukins in disease processes, you'll have this framework to then build on. And that's the whole purpose of understanding these concepts in these first three lectures, is to have that strong foundation. So now we have interferons, and they are especially important for antiviral defense. The name is actually really helpful because they interfere with viral processes. They can alter gene expressions within cells, inhibit proliferation, and enhance immune effector functions. So, interferons are another good example of the immune system communicating information to surrounding cells. So, those cells can change their behavior in response to a specific threat. Then our tumor necrosis factor, or TNF, is an important mediator of acute inflammation. It helps recruit neutrophils and monocytes and promotes activation of those phagocytic cells. At an appropriate level, that's very useful. We need inflammatory cells brought to an area of infection, right? But excessive inflammation can become really harmful. And that, again, is very important throughout immunology. Um, and that's where we'll see that the same mechanisms that protect us can also cause diseases when they're excessive or inappropriate or poorly regulated. And TNF is a really great example of that principle as a whole. So, not every soluble mediator has the same job. Colony stimulating factors help regulate the production and development of blood cells. Chemokines are especially important for directing cell movement, especially helping to tell leukocytes where to go. And other growth factors regulate proliferation, differentiation, and tissue responses. This gives us another connection to hematology, right? During infection or inflammation, changes in cytokine or growth factor signaling can contribute to the changes we eventually see in the peripheral blood. All right, so now we're bringing all of this back to the laboratory. We've talked about what happens biologically. Now, the next question is, how can we detect evidence that an inflammatory response is occurring? And one answer is through acute phase reactants or acute phase proteins. Acute phase proteins change the response to inflammation, infection, trauma, surgery, malignancy, and other physiologic stresses. One of the most familiar is C reactive protein, or CRP. An important point is that different proteins respond at different rates. So CRP can rise relatively quickly, approximately 6 to 10 hours, whereas fibrinogen responds later, around 24 hours. And then C3 responds about 48 to 72 hours, and so on. So, when interpreting an inflammatory marker, the timing really matters. A laboratory value is just a snapshot of the biological process. If we don't consider where the patient is within that process, then those values can be misinterpreted to what the actual snapshot means. So that's another reason why our job is so important. So this is where I want you to really start thinking like a laboratorian rather than just simply memorizing immunology. A soluble mediator result rarely gives us a diagnosis by its own. Instead, it's contributing to patterns. So the questions that should be asked is there evidence of inflammation? Are complement proteins increased or decreased? If they're decreased, could complement activity, could complement be actively consumed? Could there be an inherited deficiency? Does the patient's history fit an immune complex process? And then the most important question is what additional testing would help clarify the picture? So, all of that is laboratory reasoning. So, we're not just simply reporting numbers, but understanding what those numbers represent biologically is so important. We don't diagnose patients as laboratorians. That's beyond our scope of practice, right? But we want to understand the mechanisms behind each laboratory test. So then we understand the lab result when we get the call. What does this mean? How would you interpret this? Would you recommend any reflex testing? Then you're able to have an educated response to any and all of those types of questions you may get out when you start working the bench. So, if you remember 6 things from this lesson, remember these. 1. Complement is an amplifying protein cascade. 2. There are 3 activation pathways, but they converge around C3. 3. Complement can coat, recruit, clean up, and kill. 4. Cytokines allow immune cells to communicate and coordinate their response. 5. Acute phase proteins give us measurable evidence on inflammatory activity. And then 6. Laboratory results never exist in isolation. They need to be interpreted in context of the patient, timing, and the rest of the laboratory picture. So, if you can explain those 6 concepts rather than just memorize the names or definition, then you've really understood a major purpose of this lesson. Like I've said before, we're setting up the foundation now for later lessons that we are going to encounter. So, as always, if you have any questions, comments, concerns, please do not hesitate to reach out. I hope that this lecture and all of this information was easy to understand. But I do know and understand that this is a lot of information and it's really complicated, but I promise that I'm giving you the big picture now, and then we're going to start to eat that elephant one bite at a time. Okay? So I know that you guys have got it. Head over to Moodle to do your weekly like homework and case study to be able to apply all of the information from this lesson. And again, please don't hesitate to reach out with any questions whatsoever. Hope you guys have a great week.